Biochemistry 401 Lecture 11. Today we're going to talk about heme, how it's synthesized, how it's degraded. We're going to talk about the reactions and the regulation of those reactions. And we'll also discuss diseases that occur when the synthesis of heme goes wrong. We're going to talk about hemoglobin degradation, specifically the catabolic reactions involved in the degradation, and then the significance of serum bilirubin levels. So let's get started. Now, when most people think of the mitochondrion, they think of ATP production, and certainly ATP is produced in the mitochondrial matrix. But the mitochondrion is used for a lot more than just that. In fact, many of the reactions that we're going to see this semester occur in the mitochondrion. Let's look at it a little more closely. The mitochondrion has two membranes, the outer mitochondrial membrane and the inner mitochondrial membrane. Just inside the inner mitochondrial membrane is a portion of the mitochondria known as a matrix. This is the site of many biosynthetic and catabolic reactions, including some of the reactions in heme synthesis. Heme synthesis consists of forming an asymmetric ring called protoporphyrin IX. Following the synthesis of this asymmetric ring structure, iron is chelated into the center. Now, the formation of heme requires 4 enzymes in the mitochondria and 4 enzymes in the cytosol. Reaction 1 is a condensation reaction that occurs in the mitochondrial matrix. This is a condensation between succinyl-CoA and glycine to form delta aminolevulinate. This is abbreviated ALA. The enzyme that carries out this reaction is delta aminolevulinate synthase, and it happens in the mitochondrial matrix. One of the reactants, succinyl-CoA, is a product of the TCA cycle that occurs in the mitochondrial matrix. We're going to talk about the TCA cycle later on in the course. And so let's look at this reaction a little more closely. This reaction involves the condensation between succinyl-CoA and glycine. Now, succinyl-CoA is an activated form of succinate. Generally, when we look at condensation reactions, we're going to have at least one of the reactants that is activated to help the reaction proceed forward. The liberation of coenzyme A and carbon dioxide help to drive this reaction forward. The thing about this reaction is that it's a rate-limiting step. This means it is the step that takes the longest time and dictates how fast or how slow the whole pathway will go. Now, the important thing about this is that generally the rate-limiting step will be the most highly regulated step in the whole pathway. No sense in taking a whole lot of time in synthesizing something if you don't really need it. This step takes place in the mitochondrial matrix, and its regulation occurs by negative feedback through heme. First of all, heme can inhibit the transport of delta ALA synthase into the mitochondrion. Delta ALA synthase is synthesized in the cytosol, but it works in the mitochondrial matrix. So therefore, if you can block its entry from the cytosol to the matrix, you can block its action. Heme also has a negative effect on the transcription of delta ALA synthase, on the message's stability, and on its translation. Delta ALA synthase is a very short-lived protein and is rapidly degraded within 30 minutes of its synthesis. So unless there is continued transcription and translation, the concentrations of delta ALA synthase in the cell will decrease. The next reaction to occur is a dehydration synthesis. First of all, delta aminolevulinate is transported to the cytosol, and then 2 molecules of delta aminolevulinate will condense to form each porphobilinogen molecule. It takes 8 molecules of delta ALA to make 4 porphobilinogen, and we need 4 porphobilinogens for the next reaction. The next reaction, reaction 3, is a condensation reaction. We're going to link 4 molecules of porphobilinogen head to tail to form a linear tetrapyryl molecule. This occurs with the release of an ammonium ion for each methylene bridge that's being made. The enzyme that does this is porphobilinogen deaminase. So now that we have a linear tetrapyryl, we're going to cyclize that to form an asymmetric ring called uroporphyrogen 3. The enzyme that catalyzes this is uroporphyrogen 3 synthase. Now, if you'll notice, there are 2 types of substituents that are linked to this ring, acetate and propionate, designated as A or P. If you'll notice the portion of the ring that's highlighted in orange, you can see that this is different with respect to the substituent's location, while the other pyrrols in the ring are acetate, propionate, acetate, propionate, acetate, propionate. The pyrrole that is highlighted in orange is propionate acetate. This ring has been flipped in the opposite orientation, ensuring that the ring that's constructed is an asymmetric ring. This is important because uroporphyrogen 3 can be used in other reactions in the pathway, whereas a symmetric ring, one that does not have this 4th pyrrole flipped, will be a blind alley. A symmetric ring will not be used in further reactions and builds up as a waste product. We're going to look at this a little more closely later on in the lecture. But it's important to know that the enzyme uroporphyrogen 3 synthase constructs an asymmetric ring from the linear tetrapyryl. This asymmetric ring that's constructed is uroporphyrogen 3. The next reactions alter the substituents of the ring to form methyl and vinyl side chains, in addition to the propionate ones that are already there. Additionally, the saturation of the ring is also changed. And finally, one ends up with protoporphyrin 9. These reorganizational reactions occur in the inner membrane of the mitochondrion. And finally, protoporphyrin 9 is transported to the mitochondrial matrix, where iron is chelated. The final step is the chelation of ferrous iron, Fe2+. Now what chelation is when multiple bonds are made between a central metal and another group. In this case, the other group consists of 4 nitrogens, each of which is a substituent of one of the pyrrole rings. This ferrous iron is positioned by the enzyme ferrochelatase. Problems with the heme synthetic pathway are known as porphyrias, and there are 2 main types. These are acute intermittent porphyria and congenital erythropoietic porphyria, and each of these is caused by a deficiency in a particular enzyme. Acute intermittent porphyria is a deficiency in the enzyme that catalyzes reaction 3. This is a porphobilinogen deaminase deficiency. In this case, porphobilinogen and delta-aminolevulinate are found in excess, and this is because there is a logjam at reaction 3. The pathway cannot proceed to make the finished product, heme, and in turn, because heme concentrations are low, the negative feedback loop that would turn off the first reactions is not there. And so there is a buildup of these intermediates, delta-aminolevulinate and porphobilinogen, Because of the excess of these metabolites, the following symptoms are found: severe abdominal pain and neurologic involvement. It is thought that the neurologic involvement might be because aminolevulinate looks a lot like GABA, which is a neurotransmitter. It is thought that King George III had acute intermittent porphyria, and that this accounted for some of his more radical decisions. On the other hand, in congenital erythropoietic porphyria, we have a deficiency in the enzyme that catalyzes reaction 4. This is a deficiency in uroporphyrin 3. This causes uroporphyrin 1, which is a symmetric ring shown on the right, to be formed in addition to uroporphyrin 3. Now the thing is that uroporphyrin 1 has no use. This ring and its metabolites build up. They build up in the enamel of the teeth and under the skin. Now, because this is a conjugated system of interspersed single and double bonds, this is a molecule that can absorb and emit light, and it gives rise to a red color, the same color that one sees in blood. Unfortunately, because this ring absorbs light under the skin, it causes lesions of the skin, photosensitivity, and it also can cause red fluorescent teeth. It is thought that vampires were first attributed to persons who had congenital erythropoietic porphyria because of the photosensitivity and because they had red fluorescent teeth. In an effort to explain these symptoms, individuals were seen as being markedly different and having a supernatural component. Now that we've looked at how heme is made, let's look at how heme is catabolized. As we said before, red blood cells circulate for an average of 120 days in the bloodstream, and they are filtered out of the bloodstream by the spleen. The hemoglobin protein components, the globin chains, are hydrolyzed to individual amino acids. The iron is recycled, and the heme is broken down in several steps to form one stercobilin, which is excreted in the feces, and one urobilin, which is excreted in the urine. Let's look at this pathway. The first thing we're going to do is we're going to break the alpha-methane bridge that is shown by the red arrow. This is done by heme oxygenase. When this happens, we have a release of ferric iron, and the products of this reaction are a small molecule called biliverdin, carbon monoxide, water, and NADP+. The reaction in which heme is broken down to form biliverdin requires an electron carrier NADPH. This electron carrier is going to donate electrons to the heme ring in an effort to split it. Once we have biliverdin, we're going to then reduce it through the action of NADPH again to form bilirubin. NADPH will be oxidized and biliverdin will be reduced to form bilirubin. And the sites of reduction are shown in blue in the figure below. The enzyme that catalyzes this reaction is called biliverdin reductase. Again, this is because biliverdin is being reduced. And we have a reduction of the central methane bridge. Again, here we see the same reaction that's catalyzed by biliverdin reductase. Sometimes it helps to see things in a different way so that we can see where the sites of reduction really are. Again, NADPH is used as the electron donor, and it is oxidized to form NADP+, whereas biliverdin is reduced to form bilirubin. Subsequent reactions necessary for catabolism of bilirubin happen in the liver. The thing is, bilirubin is not very hydrophilic, and so to be transported in the blood, it needs a carrier, and this is serum albumin. Serum albumin binds to bilirubin and to other poorly soluble molecules to transport them through the bloodstream. Once bilirubin reaches the liver, a carbohydrate derivative is added to make bilirubin more soluble. In this slide, we see an activated acidic derivative of glucose, UDP glucuronide. We will see many examples of carbohydrates that are activated by dinucleotides in synthesis reactions. As we have seen before, in order to make a covalent linkage between two metabolites, one of the reactants must be activated. And in this case, the sugar was activated by UTP to form UDP glucuronide. The enzyme that catalyzes the transfer of glucuronide to bilirubin is called glucuronil transferase, and this is a liver enzyme. It's going to transfer two activated glucuronide sugars, UDP glucuronide, to the two propionate side chains of bilirubin to make bilirubin diglucuronide. This is also known as conjugated bilirubin. And now this metabolite is hydrophilic enough to be transported in bile through the bile duct to the small intestine for further processing. And so before we go any further, let's go over again the differences between unconjugated and conjugated bilirubin. First of all, we start out with bilirubin that's formed in the spleen from biliverdin. Bilirubin then is bound by serum albumin for transport in the bloodstream to the liver. Once in the liver, glucuronil transferase transfers two molecules of glucuronic acid to two propionate side chains to form bilirubin diglucuronide. This is conjugated bilirubin. This conjugated bilirubin is then transported in the bile through the bile duct to the gallbladder and then to the small intestine for further processing. Once in the intestine, the glucuronides are removed and further breakdown occurs through processes that are carried out actually by the gut bacteria. The product of this breakdown is 1-urobilinogen. This has an orange-yellow color. Some of this is reabsorbed and transported to the kidneys, where it is excreted in the urine. The remaining product is broken down further to 1-stercobilin. This is more of an orange-red color and is excreted in the feces. Now, just as there can be problems with the synthesis of heme, there can also be problems with degradation, and there are tests to figure out exactly where the problem is coming from. There are two types of tests, one that tests for unconjugated bilirubin and one that tests for conjugated bilirubin. An indirect test of serum bilirubin tests for the unconjugated bilirubin. Now this is bilirubin that has not gotten to the liver yet. It's not conjugated. An increased concentration of unconjugated bilirubin can be the result of anemia, neonatal jaundice, or the resolution of a large hematoma. An increase in the levels of unconjugated bilirubin is a signal that the body is trying to get rid of a lot of heme at once and is overwhelming the system. But this is generally not caused by liver damage. Rather, it's caused by an increase in red blood cell lysis. Now, as we saw with sickle cell anemia, anemias can result from the rupturing of red blood cells. And when this happens, a lot of heme must be processed. In neonatal jaundice, a lot of bilirubin must also be processed at once, and bilirubin levels can build up extensively in the tissues. Now, this can have a really bad effect on brain functioning and should be combated quickly. And this is done with UV therapy. Babies will be put under UV lights, and the reason for this is bilirubin can undergo a conformational change when exposed to light that results in its ability to be excreted more easily. An interesting note is that the reason that bruises change color, going from red to green to yellow, is the process of going from heme to biliverdin, which is green, to bilirubin, which is yellow. On the other hand, a test for serum bilirubin that detects conjugated bilirubin looks at bilirubin that has been processed by the liver. When this builds up, that means that there is a problem with the liver itself. And this happens when the liver cannot dispose of this normally through the bile because of bile duct obstruction, because of cirrhosis of the liver, or because of hepatitis, which is an infection of the liver. Now, total serum bilirubin measures both indirect, unconjugated, and direct, conjugated bilirubin. And to figure out the differing components, one subtracts the direct from the total to get the indirect. And that's it for Lecture 11. Have a great day.